I'd forgotten that you had muted me as well as everyone else. Sorry about that.
And Tom, you're muted. You'll need to unmute. There you are. Perfect.
Very good. Oh,
Right.
I see, right. Manual unmute.
I'll start again. So welcome everyone to the Exeter Beekeeping presentation this evening by Professor Tom Seeley, titled Darwinian Beekeeping. My first vote of thanks has to go to our Secretary Kathy Mudge for organising this presentation by Tom. What an achievement she's made for us. You will all know that Tom is one of the most knowledgeable people in the world in the study of honeybees, if not the most knowledgeable, and hence we are immensely privileged to hear from Tom directly. However, it's still worth reminding ourselves on the scale and recognition of that knowledge. Tom's formal title is the Horace White Professor in Biology at Cornell University. Before that, he held positions at two of America's most prestigious universities of Harvard and Yale. Tom also spent several years at Berlin and Würzburg universities, so has a thorough appreciation of European beekeeping. Tom has written 5 books on the honeybee and received numerous awards for his research. His books include The Wisdom of the Hive, for which he received the gold medal for the best science book of that year. This was later followed by the very well-known Honeybee Democracy in 2010. Other accolades have included being elected a fellow of several prestigious organizations, such as Fellow of the American Academy of Arts and Science, Fellow of the Animal Behavior Society, and more recently, Fellow of the American Association for the Advancement of Science. However, Tom says that his most enduring honor is to have a species of bee named after him, which is, I'm probably going to mispronounce this, Neocorinurella seeley.
Very good, very good.
I repeat, we're immensely privileged to be able to hear from Tom directly, and so listen well and enjoy the experience. Thank you.
Thank you, Basil. Let me just check with Kathy. Do you hear me okay, Kathy?
Yeah, we can all hear you, Tom.
Okay, very good. I'll switch over. I'll pull up this. I'll share, yep, and then there. That should do it. There we go. Great.
Sorry, Tom, before you start, I forgot to add a bit. And please, people, Tom has asked that if you want to ask questions, you put them on the chat facility on the bottom, and Tom's going to make a couple of breaks as we go through those questions. Sorry, I should have mentioned that earlier. Apologies.
Thank you. Thanks again. Great. One thing Basil didn't mention is that yeah, I'm a scientist, a biologist, but I'm also a beekeeper. I've been a beekeeper since I was since I was a teenager. And that's what underlies my scientific work is my fascination and appreciation and really love of honeybee colonies. I think they're just an amazing piece of biology. Well, what we're going to look at today is this topic that I call Darwinian beekeeping, or which briefly described is an evolutionary approach to apiculture. And I would like to start this talk out by showing these two photos. Well, one's a photo and one's a painting. You probably recognize these two gentlemen who look somewhat similar, and that's because they were similar in a number of ways. On the left, Lorenzo Langstroth, on the right, Charles Robert Darwin. They were
Tom,
similar.
sorry, we can't see those pictures, so I think you need to do a share screen, please.
Oh, I think I did. I thought I did that. Okay, let's go back then. And that. Let's see, down at the bottom. Oh, I know. Yep, I had things slid down. I need to share screen. This will work much better. There we go. So now you're seeing my screen, and I need to bring up the video or the PowerPoint, which is, that's hiding down here. And bring it up. Okay, do you see things now?
Yep, that's working perfectly now. Thank you, Tom.
Okay, great. I'll just repeat. Here we see the title again, Darwinian Beekeeping. And you'll see these two gentlemen here. And before I reveal who they are, I guess I have already done that. They are Lorenzo Langstroth and Charles Robert Darwin. And you can see they were essentially contemporaries, born almost the same year. And the reason I've juxtaposed them is that they both had a keen interest in honeybees and made important contributions to our understanding of honeybees. Through Langstroth, was the invention of the movable frame hive. That was an outgrowth of his studies of their comb building behavior. And for Darwin, it was an admiration of their comb building behavior. One of the challenges he faced with his ideas about evolution by natural selection was, how could evolution produce bees with the skill to build these wonderful wonderfully regular hexagonal cell combs? And he realized that when he looked at how other less social bees build their cells in their nests, he realized it's just a matter of building cylinders of the same size close together, and they will adopt that hexagonal shape. But they had some other things in common. And the one I want to emphasize today is that both of these gentlemen had insights that can help us with our beekeeping. For Langstroth, it was the concept of the movable frame hive, and for Darwin, it is his concept of evolution by natural selection. And in case, well, I want to make sure everybody's clear about what I mean when I say Darwin's concept of evolution by natural selection. And I want to illustrate it or review it using the well-known example of the evolution of resistance to pesticides in insects. And the way this resistance evolves through natural selection is illustrated in this diagram. You could have a population of individual insects before selection that vary in their resistance level, color-coded here, from low to high. So you got a range of resistance levels before selection. Then the pesticide is applied in a population or over an area, and only the individuals that have high resistance survive. And then as the population rebuilds itself, it will be composed of individuals with high resistance. So that's how evolution by natural selection works. Selection takes out individuals that don't have the means to survive and reproduce in a particular situation. So it's a very, very straightforward idea, but it's a very profound idea and it has widespread significance. What we're going to focus on now is how this concept of natural selection applies to beekeeping. You might be wondering, how so? Well, I'm going to make a strong statement here, but it's true. Darwin's concept of natural selection tells us that everything that colonies do when they're living on their own is done to favor their survival and their reproduction. That's what natural selection favors, is the traits that enable organisms to survive and reproduce. And I've put into bright white letters here the word everything, because I really mean everything. And everything that you can think of inside a bee colony is there because when one way or another, it helps that colony to survive and reproduce. Of course, with the efficient structuring of their hexagonal cells, the way the queen lays an egg so that one end is glued to the bottom of the cell and the rest projects up, the way the nurse bees, put in plenty of brood food for the larvae, or the structure of the bees themselves, their beautiful pollen collection apparatus, their wonderful tongue, their whole flight apparatus, everything, their amazing sensory abilities, all of that is there because those are traits or structures that enable these bees to survive and reproduce. In the case of bees, it's survival and reproduction of their colony. So everything the colonies do when they're living on their own, and when I say that's a key point, when they're on their own, when they're controlling their lives, is done to favor their survival and reproduction. And one way to put this in a few words is to say the bees are superb beekeepers. They know how to keep themselves very well. And the heart of Darwinian beekeeping is allowing the bees to use their own beekeeping skills fully. Sometimes this is called natural beekeeping or apicentric beekeeping, but I like to, because it's grounded, the perspective I'm promoting is grounded in Darwin's own ideas, I like to give him due recognition. So I call it Darwinian beekeeping. And one thing I want to stress at this point is Darwinian beekeeping is not for everyone. You'll see why very quickly. But in essence, it's good for somebody who keeps just a small number of hives. But before we get into that, I want to address a basic question. Does it make sense to allow the bees to be their own beekeepers? Don't honeybees these days need our help to deal with varroa? And I certainly used to think that was the case. And it's probably the case in some places, but it's not the case always. And what I want to do is I want to share with you a place where I'm studying honeybee colonies that are living wild. That is to say, they're living on their own up in trees, and they don't need varroa treatments. They're thriving without all on their own. Where is this place? And so I just make this point. Wild colonies in trees and buildings are showing us, have taught me that bees don't always need our help to deal with varroa. The place that, and I learned this back in 2002, and that's when I learned that the unmanaged colonies, the wild colonies living in trees and buildings around Ithaca, New York, have not been killed off. They're still there. They're still thriving. Let me give you an example. The example is from the forest areas outside of Ithaca. And here's a map just to help everybody get oriented. It's a map of New York State. New York City is down here at the lower southeast corner. And I don't have a scale on this map, but if you wanted to drive from New York City up to Ithaca, you've got about a 5-hour drive. So I don't know what that's like in England from Exeter. That would get you up to beyond Birmingham, maybe, maybe farther, maybe even to Edinburgh. Now, it's a 5-hour drive. And the other thing I want to stress on, show on this map is that the area, we're in the foothills, the uplands of the Appalachian Mountains. So I'll show you shortly that there's wild areas, very heavily forested wild places to the south of Ithaca. It's an area with extreme climates, hot summers and cold winters. And here's a Google Earth view that zooms us in a little closer to the place that lies at the heart of this talk, which is the Arnott Forest. Here's Ithaca at the southern end of this lake called Cayuga Lake. All these lakes were riven out by the last time glaciers passed through here about 10,000 years ago. And they only went so far though. And if you go south of Ithaca, you get up into the hills and you can see these hilly places south of Ithaca are covered with forests, with trees. And one area that is special to this talk is the area called the Arnott Forest. I've been studying the wild colonies living here in this forest since 1978. This is a forest that's owned by Cornell University. And over the, what does that make it? It's 22 plus 21. Over the 43 years I've been watching the bees and this forest, I've been watching the forest itself get wilder and wilder. When I started out, there weren't black bears or ravens or bobcats or fishers. It's kind of weasel, big weasel living in this forest, but now all of those animals are there. And there used to be farms up here, but in the mid 1800s, the land was abandoned because it was not good farmland. So now it's grown back to forests. Here's what it looks like when you're up on the top and you look out as far as you can see. I'm looking south up to the Appalachian Mountains. It's forest lands, forested hilltops and slopes. There are roads in the valleys and some towns and even a few cities down along the rivers, but most of these hills are just heavily covered with woods. They grow excellent hardwood trees, and that's the major industry in this area, is growing, or the lumber mills. And the reason I focused on the Arnott Forest in this talk is it's the one place in all of North America where we have data on the abundance of wild colonies, bees living on their own. Data on the wild colony abundance before the arrival of Varroa. Now, Varroa got to the United States. It came in through Florida back in the late 1980s, but it didn't reach New York State, or where I live, until about 1994. That was the first time I saw Varroa on my bees. But back before Varroa came, so 16 years earlier, I had gone to the Arnott Forest and I had mapped out the wild colonies in the forest. And I found one colony, 1.0 colonies per square kilometer. So I had baseline data on the abundance of wild colonies in this forest long before Varroa arrived. In the mid-1990s, I was losing about 80% of my colonies each winter to Varroa. I didn't know about Varroa and was clueless for a couple of years. So by the year 2002, I wondered, are there any colonies left in that Arnott Forest? Nobody is giving those bees miticide treatments. Can there be any colonies still there? So in 2002, I went back to the Arnott Forest and repeated my bee hunting, which is the process I'll talk about now. This is how I find wild colonies living deep in a forest. I won't go through it in detail, but I'll tell you a little bit about it in a nutshell. The bee hunting goes as follows. You go to a field where there's flowers, wild flowers with bees. You capture some bees in a little box called a bee box. Once you've caught some bees in the box, you can add a square of comb filled with sugar syrup, and you introduce the bees to a wonderful food source. The bees that have been introduced to it, they bring their hive mates or colony mates back to the feeder, and soon you can have a pretty heavy traffic of bees to and from their hidden home and your little feeder comb. And then what you do is you see in what direction the bees fly home. Sometimes you'll get two bee lines, but you choose one and you determine their bee line. That will be the focus of your interest. And then what you do is, and I'll illustrate this with a bee hunt that I did on the Cornell University campus. You then move your little feeder with the bees step by step off in the direction they're going. So here's a bee hunt where I started at noon, caught bees on flowers in a garden here, and saw that they were going sort of up in this direction. So I moved the feeder over here to get it away from this tall building. And then I could see, oh, these bees are really going almost due north. And then so about every hour I moved my feeding station with the bees, carrying them along from here to here to here to here to here. When I got here, I could see that I was getting close because I have some of the bees labeled with paint marks, and I could see it was only taking those bees about 3 minutes to leave my feeder, go to their home, offload their nectar, and then return to the feeder. So at that point I said, okay, I don't need to get any closer. I know the direction to that tree. It's not too far away, within 100 meters. And when I went down, searched along the line, that's what I found. I found the bees flying in and out of a knothole up in the top of this black oak. You might wonder, well, how do you see bees flying in and out of a knothole that high up? Well, it's not so hard because I hope this video comes through. What you can see from the ground, even if the nest is high off the ground, is the flight activity of the bees. You look up and you see these little dark blobs zooming around up there, and that's how you can spot the nest entrance. It's not as hard as it sounds. So here's what I did in 2002. I went to the Arnott Forest. Here's the outline. It's a jagged boundary. And I started in the back entrance of the Arnott Forest. I set up caught bees off the flowers. I let them off, let them fly home. And I soon found that there were bees coming to my feeder. I had captured bees from two wild colonies, one to the south and the one to the north. So it took me 2 days to work my way down the line and find this tree. Once I found it, I went back here, picked up the bees, picked up the line that it was bees that were still interested in it, going to the north, found that one. Then I shut down the feeder here and started up with a new bunch of bees over here. And from here, I got bees flying off to this tree A, the first one, so I didn't have to look for that nest again. But some of the bees now were going up in this direction. So I lined my way to that tree. And basically, I continued that process around the western half of the forest. And I found, let's see, what is it? It's 8 colonies in the in that side of the forest, west side of the forest. So it was clear that, and I was surprised but delighted to discover there were still wild colonies of bees living in this forest. Oh, and here's a picture of what one of these trees that's housing a bee colony looks like. That's me at the bottom, so you can see this is a big tree. It's a tall tree, and the bees are, their entrance is way up here. And again, one of the hardest things about finding a bee tree is that last step. Sometimes it's just very hard to spot the opening where these bees are flying in and out. But this one was handy. They were right, it's a sunny spot. You could see the bees. I was able to see the bees easily. If a little plug for a book, if you're interested in this fascinating art of skill of working with bees called bee hunting, I wrote a little book on it. It's so much fun to do. I said, this is too good to not share with others. So I wrote this book called Following the Wild Bees. So, as I mentioned before, the Arnott Forest is the one place in North America where we had this data on the abundance of wild colonies before, and now we have it after the arrival of Varroa. And it was the same abundance, exact same abundance, 1 colony per square kilometer. Now, how could this be? Well, I thought either these bees are really good at controlling the Varroa, or maybe they don't have Varroa. At this point, I didn't know if these colonies had Varroa because they're living up in their tree cavities, and I can't go in and sample the combs and look at the brood or anything like that. So what I had to do was to answer this question, do they have Varroa? I put up bait hives in the forest. I would take old Langstroth hives and I'd nail some boards to support them up in the tree, up on a tree. And the reason I put them up high is I know that's what nest site scouts like in a home. They like their nests to be high. I could talk for a long time about why bees like their nests high up, but I can summarize it in 2 words, black bears. It really, if there's a colony near the ground, the black bears are all over it. But up high, the bears don't find them very well. So I was putting my bait hives up off the ground. I make the entrance, give it a small entrance. I block off most of the entrance because I know the wild colonies and bees like small entrances, and I'd face it south. And so I put these up, and over the next 2 years, I caught 11 swarms in the Arnott Forest. And now that I had bees living in a movable frame hives, I could check them for Varroa and other diseases. And every colony, all 11 colonies had Varroa, came in with Varroa. One colony developed chalk brood, but none of the other fowl broods, neither of the fowl broods was detected in those colonies. But the key point here was the Arnott Forest colonies were all infested with Varroa. And yet they were surviving, and they're still surviving. Now, how can that be? So to come back to this question, can we let the bees be beekeepers? The answer is yes, we can, at least in some places. And I think what I'd like to do at this point is stop and check in and see what questions people might have so far. Because what we've looked at so far is maybe showing you that there are, in places where colonies can live wild, there can be wild colonies. And as we'll see shortly, they know all about Varroa, how to deal with Varroa. So remind me how I get questions. So I think I go down to the bottom here. Oh yeah. So let's see. Basil, do you want to take questions and announce them, or should we proceed?
Yes, Tom, I'm happy to do that. Right, we've now got a couple of questions. Right, we've got one come in from Rose says, what is the risk of varroa if left untreated infecting bumblebees?
Oh, I don't think there's much risk at all. Varroa is very much an honeybee apis mellifera specialist.
Okay, that's the only one we have at the moment, I'm afraid, Tom. So I think it's
Okay, great.
people are still thinking about what you're telling them.
I have
Okay,
a question that I'm just typing in, please, if I may.
Sure, thank you, Chris. Yeah.
Were the colonies that you found, Tom, were they heavily infested with varroa or only lightly infested?
I'll come back to that. I think I've got that in this talk.
Okay.
They would grow. They would become in the spring. They would have very low levels of varroa, and it would gradually rise in the summer, but never got to a fatal level. Didn't get to fatal levels.
Thanks.
Yep, that's an important point. Thank you for asking that question.
And there's another one that's come up from Ed Maltby, Tom. Do wild colonies remain in place, or are the sites abandoned from time to time?
There is turnover in the locations, in the sites, because every year we'll look at this near the end of the talk. About 20% of the colonies die every winter, and that of the established colonies, and those colonies are replaced by swarms the next year. But they don't. But a cavity can sometimes gets reoccupied. Where a colony dies out, the cavity sometimes get reoccupied and sometimes not. So that's why the bee hunting has to be done every few years, because the locations where the bees are changes.
Okay, and that seems to lead nicely on to the question from Karen. Hi, Tom, your map seemed to show that there were some colonies that you didn't find. Could the density of colonies be higher than the 1 per kilometre?
Let me go back to that question, because I think I didn't probably state it clearly. Yeah, this eastern portion of the forest, I didn't search. It's to find it took all of the time that I had available that fall to search the western half of the forest. So there is this whole area over here, which it's blank in terms of bee trees, but simply because I didn't have time to search that area. That's another important question.
And then the final question for this session, and we've got one from Graham. 20 years of treatment impedes Darwinian beekeeping, but allows varroa to race ahead. Who wins?
Varroa. Yeah, that's an important question too, because what I'm advocating is if you're in the right location and you're of the right size operation, you can actually, you may want to consider not treating your bees and taking some knocks for your bees, but you'll come out at the end okay. And I know, because I've been there and I've done that, and that's what I'll be talking about.
Thank you.
Yep, I guess I should go on at this point. I've said that Darwinian beekeeping is a process in which you let the bees be their own keepers. And what I want to do is develop these ideas more fully now. And again, a couple of things I want to make sure I explain clearly at the outset. This is not an approach to beekeeping for large-scale beekeepers. A large-scale beekeeper has so many hives, he or she has to keep for example, has to keep those colonies pretty close together just to be efficient in the management of those colonies. And it may not be suitable for urban beekeepers, where a person, if they want to have more than a couple of colonies, they're going to have to keep several colonies living close together. But it can be done in an urban setting. There's a wonderful book that came out recently called The Idle Beekeeper. It's by a gentleman in London who keeps 2 colonies up on the top of the roof of his house. And he is a Darwinian beekeeper. He keeps his bees in a certain kind of hive, and he puts takes off a honey super every fall and puts on an empty super at the bottom before the winter starts. And that's all he does. And he's a Darwinian beekeeper. Anyhow, Darwinian beekeeping is an option for small scale beekeepers who want to avoid chemical treatments and who are satisfied with a modest honey crop, maybe about 25 pounds per hive. Do you guys use pounds or kilograms these days, or both? Now, another way I want to put Darwinian beekeeping in context is to say is to contrast it with commercial beekeeping, which I started to do, but it's a lot like the different, and that difference between Darwinian beekeeping and commercial beekeeping is a lot like the difference between having a one backyard apple tree versus having a commercial orchard. Or it's even better analogy is the difference between being a bird watcher and a poultry farmer. When you're a bird watcher, what do? You put out maybe nest boxes for the birds, and you set up a bird feeding station. You just enjoy watching the bees and having them part of your life. You're not trying to make a living out of it. And so one, Darwinian beekeeping is really all about pleasure with the bees. It's not about profit with the bees, though you can, on the right, if you get in the right location with a suitable scale, you can still make some money from the honey. But it's not a big commercial process. It's not suitable for large-scale commercial beekeeping. But maybe, so I hope that's clear, but I also hope that it means that most of the folks in the audience are more like the bird watcher, really into the bees, particularly for pleasure. And again, I want to emphasize both approaches are valid, they just have different aims. But in my experience, when I was taught beekeeping, the only beekeeping I was taught was the kind of beekeeping that was done for a commercial beekeeper to maximize honey production and minimize swarming and really do a lot of manipulations of the lives of bees. So this Darwinian beekeeping, again, is about letting the bees control their lives and enjoying the bees. Now, here's an important distinction for understanding the basic logic of this approach to beekeeping, this Darwinian beekeeping. It's to recognize that there's two kinds of environments that honeybees can live in. One is their original environment. That's the environment in which wild colonies live, where they choose their nest site. The nest is high off the ground. It's a small nest cavity. A lot of things, as we'll look at more in detail shortly. But the alternative is the current circumstances. This is the environment in which managed colonies live. It's where colonies tend to be crowded together and have very large nests, and they put up very large honey stores. They have very large populations. So what we'll do next is we're going to go through in some detail to look at the difference between the original environment and the current circumstances of honeybees. And again, bring it back to Darwinian beekeeping. The essence of Darwinian beekeeping is to allow managed colonies, insofar as possible, to live again in their original environment. So we're going to try to put them back into their way of life, their environment that comes naturally to them, and thus allows them to use all of their skills and not burden them with greater problems. So let's go through it. I'm going to go through about 8 points of comparison between original environment and current circumstances. The first one is a really important one. In their original environment, wherever honeybees lived on planet Earth, originally, the colonies are, well, wherever they and still are genetically adapted to their location. They've gone through many thousands of years of natural selection. So the genes that work in that environment are the ones that the bees have. But in current circumstances, that's not always the case. Colonies are often not genetically adapted to their location. And I don't know how this works in the UK, but in the United States, it's glaring. Oftentimes, bees are shipped in from the south or other places to northern states where the winters are very harsh, and the bees are just horrible at dealing with northern climates if they grew up in southern climates. And here's probably an ultimate example of the mismatch is when this is from an ad in one of the bee journals here, the ultimate queen bee from Hawaii, from the Hawaiian Queen Honey Company. Well, those bees produced in Hawaii have been selected for the queen producers to be excellent bees under Hawaiian conditions. But they're not excellent bees over most of the United States, because most of the United States doesn't have a tropical climate like Hawaii. And this problem applies to a lot of the queen producers in the United States, because most of them, but not all, most of them are in the southern states where they have an early season and you can start rearing queens early. what's another point of comparison between original environment and current circumstances? Well, originally colonies, and still in the wild, colonies live widely spaced out in the woods. Current circumstances, we generally crowd our colonies in apiaries. And this is an apiary that, one of my apiaries from about 20 years ago, where I had my colonies pressed right up together. Part of that was because I had to put a bear fence around the apiary. And so those colonies were living very close together. And we'll see in the woods, they're about, we saw in the Arnott Forest on average, I didn't mention this, they're about 1,000 meters apart, kilometer apart. And what do you see in my bee yard? Less than a meter in many cases. Here's another point of contrast, the size of the nest cavity. In the wild, colonies live in small nest cavities. And here's a distribution of the nest cavity volumes of 21 wild colonies whose nests, which I collected and measured the volume of their nests. And I can show it here in liters, but it's probably more meaningful to think that the units that here in terms of 10 frame deep Langstroth hive bodies. And you can see that in the wild, most of the nest cavities are somewhere around one deep 10 frame hive body. I don't know how that relates to your national hive, maybe pretty close. But very few are as large as 2, 3. There was one colony that was in a massive nest cavity. It was a massive hollow in an old beech tree, and the bees were living way up in the top of this hollow. So it wasn't like they used the 450 liters of nesting space they had, but they were in a massive cavity. Whereas in contrast, in a lot of beekeeping situations, the colonies live in supersized nest cavities. And here's a photo that I lifted from one of the bee supply catalogs. And I love this, the best and most complete wooden kit. And it's very complete. It has 2 deep hive bodies, and then it's got 1, 2, 3, 4 medium honey supers. And it's a monster of I like to say it's a supersized nest cavity, because the bees in nature would live just in that bottom box, not all of this other stuff. And we'll see some of the problems that arise from giving them such a large hive. Well, they have a massive brood nest. It's like a gold mine for Varroa, so much brood. And it also means the colonies will swarm only rarely. And we'll see too that swarming and the brood break that comes with swarming is very valuable to the bees in the wild, especially with respect to controlling Varroa. So that's the third difference, small nest cavity versus supersized nest cavity. Here's the fourth difference. In nature, the nest cavity walls are thick. They're on average about 10 centimeters, 4 inches thick, sometimes even more. And they're coated with propolis. The bees coat the walls with propolis because the interior of this tree cavities is a very porous material formed by the fungal decay. And if they didn't seal that surface off with a propolis, here's what they would be living against. And I've chipped off the propolis over this area in the lower left. And that would be a great substrate for microbes to thrive in. So they seal it off with propolis. And in our hives, of course, most of our hives, at least the ones in the US, are smooth walled, and bees tend not to put propolis on them unless you install a propolis collection screen material on this over that nicely planed lumber to stimulate their propolis coating. A fifth point of comparison between original environment and current circumstances is the amount of drone comb in a colony's nest. In the wild, colonies invest 15 to 20% of their comb area to drone comb. And again, that number comes from the measurements I made when I dissected those 21 nests of wild colonies. Whereas I was taught as a beekeeper, you don't want drone comb in your nest, and so we give them this foundation with just worker-sized cells in it, and we really push the colonies to produce little drone comb and rear few drones. And that's why do we do that? Because it is true that when colonies produce thousands and thousands and thousands of drones, they do make less honey. They have fewer workers to begin with, and they have a bigger cost of maintaining all those drones. We've measured that. It's a cost of about between can't remember the numbers, but it can be up to as much as 15 pounds of honey, or even more, to rear all those drones. Here's a sixth point of comparison. In the wild, in the original environment, the bees have nests where the entrances are high off the ground. In the Arnott forest, that average height is 8 meters off the ground. In a beekeeping situation, the nests, our hives are at ground level, and we put the entrances at the bottom. And so the nests are quite low to the ground. And probably this doesn't apply to where you live because you don't have deep snowy winters, but a lot of the challenges bees have where I live with an entrances close to the ground is they have a hard time getting out on the warm sunny days in the winter, because often the entrances can be covered with snow. Beekeepers will put in a top entrance, but as we'll see, that creates another problem for the bees. They lose a lot of the heat out of their hive. Well, in nature, the bees live well above the snow, and in the winter, it actually can be warmer on a sunny day. It can be much warmer up here than it is down in the shady woods below. So they actually have good flight in the winter from these high entrances on a warm day. And of course, it makes them much more vulnerable to predators down low than up high. I don't know if you have predators. We have problems with skunks and raccoons and as well as bears. Another difference between original environment and current circumstances is that in the original environment, colonies had diverse pollen sources and still do when they live in wild places. Whereas in some places where beekeeping is done, those are highly human modified environments, and sometimes those environments have non-diverse pollen sources. The extreme example is shown here. It's the almond orchards in California. I think I've got one more point of comparison. Oh yeah, original environment. Colonies are not treated for diseases. And no surprise, as we'll see, bees evolve resistance to Varroa. The colonies that can't control Varroa die. Whereas under current circumstances, colonies are treated for diseases. And this inhibits the evolution of resistance to Varroa. Lets colonies that have no resistance persist. So, insofar as possible, our goal is to put these, for somebody who wants to pursue beekeeping, the goal is to put managed colonies back in the honeybee's original environment, their environment to which they're evolutionarily adapted. We do so to allow the managed colonies to live more naturally and use all of their skills, all of the skills that they've endowed with by evolution. And yes, the colonies will make less honey, but as we shall see, they will have better health. And it is a huge, and both of those things are very stark. Less honey, better health. I think, Basil, maybe at this point, it would be good to take another question break. What I want to do next is I want to talk about actually how you implement Darwinian beekeeping, and maybe that will come up in the questions. And if it does, then I'll say, well, we'll get to that. But maybe there are questions about just the logic and sense of Darwinian beekeeping. That would be good to cover that now, I think.
And we've got a question here from Colin saying, have bees now adapted to thrive in movable frame hives rather than trees?
I don't know whether the mobility of the comb and the way the combs are not really connected to the walls. I'm not sure if bees have adapted to that or not.
Any further questions from people, please? Right, I've now got one from David. The nearest feature to a hollow tree for many of us is a disused chimney, of which there are many in this country. So when the bees swarm, they quite regularly aim for these in our urban environment. Any comment on that?
Yeah, a couple of thoughts on that. I wonder if those are really good places for the bees to live, because they don't seem, they're probably not, I don't know if they're just, if they're masonry, they're not well insulated. I wonder if they're going to those just because that's all they can find. Um, and secondly, I'm also wondering if the chimneys are disused, does that mean that they are actually okay for the bees to live in those chimneys? I hope so.
In the UK, like you say, there would be an easy environment for them to find from where they live, and because most of us are moving towards central heating, a lot of these chimneys just wouldn't be used anymore. So yes, I think that's probably an easy environment. Also, one of the hardest places to get them out of. Right, I've got another question from Alan. How far do regular inspections of colonies affect honeybee health? Wild colonies do not get smoked or have their brood nests pulled apart.
Yeah, yeah. Um, I don't remember the details, but I do remember there is a study that showed that when a colony is pulled apart and gets smoked, um, at least for that day's honey production, it's really knocked down. Um, and if it's, especially if it's during a honey flow. So yeah, there are costs, direct costs in that sense of taking the colony, because that's of course a huge disruption. Unfortunately, I can't recall that where that paper was published. I'll have to try to think of that.
Okay, and then the next question from Colin: can commercial beekeepers survive in the future? Refarming methods and monopollination.
That's a really, um, that's a question for which I don't know the answer. I know that and it's going to be location specific. I know beekeepers in the Midwestern United States are really having great difficulties keeping their bees alive because of pesticides. This is a part of the United States where it's beautiful. It used to be that beekeepers made very good honey crops from all the forage crops that were being grown, the alfalfa, the clover, and so forth. But now there's so much pesticide used, the bees don't do well in those places. They'll often die. So yeah, there really is a conflict there. Yeah.
So I think we've seen similar things here in Europe as well, Tom, in terms of pesticides, and there have been some bans, but now there being some exceptions allowed, which people are very worried about here.
Yes.
Yeah.
Yeah.
Basil,
Okay.
you from Plymouth, I'm having problems getting a question to you. So do you mind if I ask it?
Yes, we'll do one more question from
Okay.
Mark. Yep.
With there being a lot of urban hobbyist beekeepers, especially in Plymouth, fairly densely spaced, do you think it would be a good idea if each beekeeper had less colonies, which would also possibly reduce the demand on forage available to other pollinators as well, so they have a fair share?
Yeah, that's another question that it's hard to give a general answer to, but because it will depend on the availability. I know that some urban areas are actually exceptionally favorable for honeybee colonies because they tend to be warmer, and there can often be a lot of plantings of forage-rich trees. So I think it's going to depend on the location, but in general, yes, depending on the abundance of the popularity of the beekeeping, it could be over, could be too much right now.
Okay, thank you.
Yeah. Well, I think at this point I should move along. What I want to do now in the last section is give a few guidelines for Darwinian beekeeping. And so we'll go, I think I've got about 8 of these. The first one is really basic. Keep bees that are adapted to your location. And there's several ways you can accomplish this. You can rear queens from your best survivor colonies, or you can capture swarms with bait hives in remote locations. And the reason I say in remote locations is so you're increasing your likelihood of getting bees that are well adapted to your location and not just from some beekeepers, from the hive of a beekeeper that's bought a queen from California, for example, or Hawaii. Or purchase queens from a queen breeder who produces locally adapted queens. And this is becoming more and more common in North America, and I bet also in the UK. And ideally, you'll be getting your queens from a queen breeder that doesn't treat for mites. And I really want to emphasize this point. keep bees that are adapted to your location. And I'll do that by sharing with you the results of an experiment I did the summer before last, not the summer of 2020, the summer of 2019. And in this experiment, it was very simple, but it was a comparison of colonies with commercially produced queens versus wild queens, queens that I caught by capturing swarms in bait hives. Now the queens came from a company called Oliveira's Queens in California. It's Central Valley, California. It's almost a Mediterranean climate. And my bait hives were in Ithaca, New York. And in this experiment, I started each colony out as a 2-frame nuke. And I would take 2 frames of bees and brood and food from other colonies. And I would give each of those queenless colonies, each of those queenless nukes, a queen, either the queen that I had caught earlier in the spring in a bait hive, or one of these Oliveira's queens that came in the mail. And then I left the colonies alone all summer. And I set them up, both groups of colonies were set up in the same location. In the foreground here, you see the 8 hives that were housed colonies that were received an Oliveira's queen from California. In the distance, you can see the 7 hives of colonies that received a queen caught in one of my bait hives. Now, I don't know the age of the bait hive queens, but of course the Oliveira's queens were young queens. So I just left these, set them up in June, and with 2 frames of bees and a queen, and I just let them go. And I didn't do anything to those colonies. They just sat there all summer. And until October, I came back and all the colonies were there, and they were all strong. And I measured the mite level in all of these colonies in October 2019. That's near the peak level of mites for our seasonal cycle here. And then I didn't do anything, just left them there. And then the next spring, I came back and I just saw which colonies were still alive and which ones had died out over the winter. Well, let's look at the results. And so this is a comparison of the non-local stock from California versus the local stock, which I caught in bait hives, which I put in forested areas outside of Ithaca. And I measured, with respect to mites, I measured the mites per 300 bees using the sugar shake test. That in October. Now here are the counts that I got for the colonies of the Oliveira's queens: 17, 10, 4, 24, 9, 5, 18. Here's the counts that I got for the colonies that were headed by bait hive queens: 0, 3, 22, 4, 5, 1, 1, 1. And you can see that's a big difference. Now there were, in the Oliveira's colonies, there were a couple, this one and this one, that had pretty low counts, but these other colonies, with these counts per 300 bees, those are not good counts. Anything around or above 10 is trouble, spells trouble for us. The bait hive colonies were much better. So queen counts were very different. What about the winter survival of these colonies? Well, I checked the colonies on the 7th of April, and of the colonies headed by Oliveira's queens, they were all dead, except one. One of the ones that had very low mite counts in the fall. And of the bait hive colonies, they were all alive except one. And it was that one that had a very high mate count and the mite count in the fall. So 14% survival versus 85% survival. And that, to me, is such illustrates so vividly the difference in what you get when you buy a queen that, well, from Oliveira's, it came from California. It wasn't locally adapted, and it hadn't been selected. They claim those queens are Varroa sensitive hygienic, but they've not been strongly selected for mite resistance because they treat their colonies with Varroicides regularly. The bait hive queens, they were all pretty good except for this one dud, and maybe that one dud came from a managed colony. I don't know where the swarms came from, but it's possible there's a managed colony up in the hills that I didn't know about. So anyhow, that's pretty striking. Now, it's not to say that you can't buy good queens from a commercial queen producer. There was actually a third treatment in this experiment where I used queens from a non-treatment beekeeper in Vermont named Kirk Webster, and his colonies were just like the bait hive colonies. In fact, they did a little better. They all survived. So you can get good stock, but you don't, at least when I talk to American beekeepers, you're not going to get them from the really big guys. They just crank out queens. So get locally adapted stock, and ideally stock that's not just locally adapted, but it's been exposed to natural selection to be resistant to varroa. My second guideline is house your colonies in small hives. And when I say a small hive, here's what I mean. I mean one deep 10-frame hive body, plus maybe one other honey super. It could be a shallow super, it could be a medium over a queen excluder. And this enables you to extract some honey or produce comb honey, whatever you like. And to look at the effect of hive size, I did in another experiment where I set up two groups of colonies really in the same location. There were 12 colonies in each group, and one group was on this side of this building. The other group was on another side of the building. So same building here and here. The colonies were 2 apiaries close together. In one apiary, as you can see, every colony lived in a 1-story hive. And in the other apiary, every colony lived in a 4-story hive. These are honey production colonies in the 4-story, and these were simulated wild colonies in the 1-story hives. And they were all set up at the same time. They all had queens from the same commercial queen producer. So there wasn't a genetic difference between these. And they all started out again as just a 2-frame nuke in the summer, in June. And none of these colonies, this experiment ran for 2 years, no varroa treatments. So what was the percent alive 2 years later? 17% for the colonies in the large hives, 83% were still alive in the small hive group. Here's some numbers. Here's what was going on with the mite counts in these colonies. Well, all the colonies were started in July 2019, and they started out identical in their mite counts because they were set up in pairs. Two nukes were made from the same source colony. So they started out with the same mean level of mites. And mite levels stayed low because they were small colonies throughout the summer of 2012. So in May 2013, they were all looking pretty good. They all survived winter. But then the summer of 2013, things diverged. The colonies in the small hives, their mite counts never got very high because almost all of them swarmed. Whereas the colonies that were living in the large hives and were making lots of honey, their mite counts got very, very high. And that's why only a small percentage, 70% of the large hive colonies survived that second winter, whereas 83% survived in the small hive group. So that is a really clear, to my mind, really clear difference between the effects of hive size. One thing I want to mention, in this group, there were 2 colonies that survived the mites, and they knew all about how to deal with mites. And those colonies happened to be, maybe it wasn't just happenstance, they were the nastiest colonies in the group. Those were colonies that were so bad, I just brought them back from this experiment, put them well away from my laboratory, and just let them go. I didn't want to go into them. It was such an unpleasant experience to open those hives. They would just be all over me. So I don't know if that related to their ability to kill the varroa, but they knew how to deal with the varroa, and they also knew how to deal well with defend themselves from beekeepers. Those were good bees, in some sense. But it is an interesting thing, when we select for gentleness in our bees, when queen producers or even hobby beekeepers select for gentleness, there's some indication that also means we're selecting for low defensiveness in general. Okay, third guideline, space your colonies as widely as possible. Now and I did an experiment to look at this effective spacing, and this is a typical beekeeper situation. It's what I was taught when I started beekeeping as a teenager. This is what you do. You put colonies together in a row for your convenience. You put them 2 on a same hive stand. That's for efficiency. You can work one colony from this side, the other colony from this side. And then you line them up close together and they all face south, et cetera, et cetera. So they're all in a row, tight together. So I set up one treatment group was crowded colonies, as we see here. The other group was in the same area, because here's the apiary. But I had this old field situation, field and trees and shrubs mix. And I mowed out this strip in that old field, and I put another 12 colonies, but I scattered them around in this field. Now they weren't a kilometer apart. They were only about 30 to 80 meters apart, but they were much more widely spaced than the colonies in the apiary. What did I find? And then I just let these colonies go, and they were all set up at the same time. They all lived in 2-story hives. The queens all came from the same queen breeder. What did I find? Oh, and I want to stress, the colonies were never treated for varroa, never. Again, what was their survival to the 2-year mark? Well, none of these colonies were alive after 2 years. 5 of these were still alive after 2 years. And I don't know exactly what was going on here, but my hunch is that, because I did see, and I don't have time to go through this, but in this situation, if a colony started to collapse with varroa, I would see bees from other colonies coming into it and stealing their honey, and no doubt picking up mites. So we call this a mite bomb phenomenon. It probably should better be called not a mite bomb, but a robber lure phenomenon. When colonies collapse, they lure in robbers. But evidently that was less strong among the dispersed colonies. And I think this is pretty encouraging. You don't have to disperse your colonies a kilometer apart like they are in the forest. And by the way, they're so widely spaced in the forest because the colonies, there's not that a forest setting like the are in a forest can't support large numbers of colonies. So I think they're spaced to 1 colony per square kilometer because that's what the environment there supports. This situation, which is not in a deep forest, the colonies could live fine and they could have many colonies. And it looks like if you just space them 30 meters, 50 meters apart, you get a lot of benefits. 4th guideline is to line your hives with this propolis collection screens or build them of rough lumber. And this propolis envelope, there's a lot of good work on how this propolis envelope is an antimicrobial shroud and favors the health of the bees. They're working less hard to fight diseases, microbial infections. 5th suggestion, provide your most resilient low mite count colonies with 10 to 20% drone comb. And that's to enable the colonies that have the right stuff to share their genes, to spread their genes in the area where you're keeping your bees. You want to promote the genetic success of your best colonies. I like this photo because, we often talk about drones being sort of greedy or hungry. Well, yeah, even before they're out of the cell, they're begging food. I love that photo. 6th guideline, keep the nest structure intact. The bees have organized their nests in our hives in ways that suit their needs. They've done it very well, as they do in nature. At least when I was a beekeeper, I was taught things like reversing the hive bodies, spreading out the brood combs with putting an empty frame between each brood comb to prevent swarming, things like that. But now I look at it differently. I try to put each frame back in its original location and with its original orientation without reversing brood boxes and that sort of stuff, because I know that the bees work really hard to organize their nest and keep the brood warm. And I think we can really hamper them in those things if we interfere with an organization of their nests. 7th guideline, provide just a small entrance at the bottom. And it's quite common in the United States, I don't know about in the UK, to offer to give the bees a top opening. And the logic has been that helps the bees by letting the moist, humid air go out, is ventilated out of the hive in the winter. But at the same time, I think every beekeeper knows that those bees go to great lengths to seal all the cracks and openings at the tops of their hives. The bees are working hard to avoid having top entrances, so they don't have top openings. And so I I've learned, and I'll mention a little bit more evidence about this shortly, but I think it's, if we're helping the bees or working with the bees, if we don't give them top entrances. And what I use for my Darwinian beekeeping is shown here. It's a 1-story hive. In the fall, I might have taken off a honey super if I had one there. It's just a small entrance, and I put a 1/2-inch mesh screen or mouse excluder stapled over the opening. And I put a styrofoam board above the inner cover to give better insulation. This hive is not well insulated, and I'll talk about that. I'm learning every year the importance of insulation. I wasn't doing very well when I took this photo. And the reason I stress this no top entrance and just a small bottom entrance is actually from work from the UK, from your Derek Mitchell, where he's done these experiments where he's set up colonies in insulated hives, and without or with a top entrance. And what he finds is if the colony is well insulated and there's no top entrance, there's actually quite a warm, nice warm pocket, or he calls it a heat pool, in the top of the hive. But if you use the same hive, same bees, and you open up a door up here, then that heat pool gets much smaller. So that is the rationale for avoiding providing a top entrance. And you might wonder, well, what about all that condensation in the hive? Well, what David's work has shown is that up in this heat pool area, the surfaces are too warm to condense the water. The condensation occurs lower in the hive, which is where the bees are not, or the cluster below the cluster. And I know that in the winter, I know from my colonies, they can get terribly thirsty in the winter, if for lack of, because they need water. And so the condensation in the hive is probably handy for the bees in the winter. Many of you may have seen bees struggling to collect water in the winter and getting caught out on cool afternoons or cool mornings. But the fact they're going out in such difficult conditions shows their desperation for water. And that might be because they're coming from hives where there's not sufficient condensation collecting inside the hive. And I want to mention this, we do need to give our bees well-insulated homes. I have learned, looking at the older literature, that beekeepers early in the 20th century used to routinely give their hives very good insulation because they didn't move them around. And we switched over, at least in the US, to these thin-walled hives because that's what is easy to move colony. That's what migratory beekeeping favors. It makes colonies easy to move around. But back in the day when people didn't move colonies around, they gave the bees much better hive insulation. Let's see, why did I put this photo in? Yeah, okay, that photo is actually a little out of place, but I'll come back to that. My 9th guideline is one that you probably been expecting. It is to refrain treating colonies for varroa. And this can be a difficult one for a lot of people, and it may not work for everybody. It requires super diligent beekeeping. And what I mean by that is you have to monitor the mite levels in your colonies. And if the mite level gets high, more than 10 or maybe even only 7 per 100 bees, then you've got a couple of options. One option is to euthanize the colony because before it collapses and gets robbed out. And this can be done by pouring warm, soapy water into the hive at dusk. That's not good for that colony for sure, but it saves other colonies nearby. And then you can rinse those combs off. And you do this to eliminate your non-resistant colonies, and you also enables you to avoid producing mite bombs or robber lure colonies. There is an alternative, and that is to treat the colony with miticide and requeen it with a queen of resistant stock if that's available. And yeah. The last thing I wanted to mention is you might be wondering, okay, what kind of colony survival do you get, Tom, with Darwinian beekeeping? Well, I'm going to tell you the results I've gotten using two approaches to Darwinian beekeeping. One was the full out bees in small, widely dispersed hive approach to Darwinian beekeeping. And when I say widely spaced out, I've got one colony here up against the wall of an old barn, and another one tucked over here miles away over on the edge of some woods, and another one and another on the edge of another farmland area. And so these colonies are in small hives. I put a shallow super on them to make some honey, a comb honey super above a queen excluder. And as soon as they got filled up those supers with honey, I would get those supers off so that the bees would start storing honey below, which they would need for winter. And I did this for 7 years with 10 to 12 colonies a year. And these are the survival numbers I've got in that long-term study. Almost every colony survived summer. Not every colony survived winter, and the overall survival across the whole year was about 81%. And so every spring, I'd start out with only 8 of 10 colonies, and then I would just capture more colonies with swarms, with bait hives to rebuild my numbers. So that's what I got when I worked with small, widely dispersed hives for my Darwinian beekeeping. I've also given it a shot in a different scenario, in this scenario, where I've kept bees in large hives and they're crowded in an apiary. And done this from 2017 to 2020. And I would, in this one apiary, I'd keep 16 to 70 colonies a year. And I'd get excellent summer survival, 94%. Winter survival was poor. And so the overall survivorship over the years, and this is for 3 years of data now, was 67% survival from one spring to the next. For every 100 colonies I would start in one winter, or I didn't have 100 colonies. For every 20 colonies I'd have at the start of 1 year, I would end up with 13 the following year. And it's not as good as the small dispersed hives, but it had the feature that I could make big honey crops. And I was doing this not because I wanted big honey crops. I don't need the big honey. I just take it to a beekeeper and he extracts it. But I wanted to see whether you could do Darwinian beekeeping even with a large hive. And I'd have to say the results are mixed. I don't advocate this approach because it just doesn't work. And I think the main reason it doesn't work is these colonies don't swarm. They can still swarm some, but they don't swarm very much. And they have these gigantic brood nests. So some of the colonies really get into trouble. But interestingly, not all of them. 2/3 of the bees actually can deal with the mites and can survive even under these conditions. It's not great. And again, those that can't are being winnowed out every winter. And then I replace them with colonies that I capture with my bait hives. Or I will sometimes replace the colonies by making splits from the best colonies. So there are colonies out there that can even handle Varroa without treatments, even if the colony is managed for honey production. So that's another approach to Darwinian beekeeping, but it's a tougher one. You just have more colony mortality. But what you do have in both situations is you don't have any miticides. You're not worrying about the mites. You're not trying to treat for mites. You're not putting chemicals in on the bees. You're just letting the bees be bees. So that's the end of what I have to present. So I like having the bees thank Mr. Darwin. And I also want to point out that what I've talked about in this talk is covered in far more detail in this book that I wrote 2 years ago, which is about how honeybees live in the wild and how that can inform our beekeeping. And the last chapter is about Darwinian beekeeping. So go back to this.
I've got a whole string of questions now here, Tom, so I'll try and group them. I'll go on the simple ones first, and that is, do you use open mesh floors in the States?
Many people do. I do not. And the reason I don't is because there are quite a few studies that show it doesn't really help control the Varroa. It seems like it would, but it doesn't. The data don't suggest that. But one thing that's interesting is that even in cold climates, the bees don't have trouble with those open mesh floors. I guess because of course the heat goes up, and so it doesn't look like it helps, but it doesn't look like it hurts either, from what I've seen.
Okay. The next question is related to that, but I think you've already answered that one. Sorry, I'm just looking for ones that group naturally together. Would this method work if you performed Bailey comb changes rigorously each year to enable you to run larger colonies?
Well, I don't know. I'd have to know more. I'd have to be more confident that I knew how a Bailey comb change works.
Okay.
It's not a technique that I know that we use or I've heard about. Sounds good. I admire the work of Bailey. He was a very good scientist.
Okay, then I've got a question here. Do you have a system for changing the comb?
No, I don't have a system for changing the comb, and this may sound bad, but I've got combs that I've been using for 20 plus years.
Oh, really? Oh, okay. But over here, Tom, we're all encouraged to change comb regularly.
That may be helpful, but it's one of those things that is probably has benefits in it, but it certainly also has costs to the bees. That's a lot of comb building to ask them to do.
Okay. And then I've got a question here. What is your preferred way to monitor mite levels?
My preferred way is the sugar shake or alcohol wash method, where I scoop up 300 bees in a cup measure. It's actually not a full cup measure, and then put them either with powdered sugar in an alcohol wash. That gives you a really good clear percentage. And you take the bees from the brood frames.
Okay. And I've got a question here. Is the use of a queen excluder preferable, keeping the brood nest relatively small? No queen excluder would seem more natural, but would the brood nest be unnecessarily large in relation to the provided space?
Well, that's a really interesting point. The bees know all about keeping their brood nest intact and compact, but yes, the queen excluder does have the advantage. It limits the magnitude or size of that brood nest. So I can see that's an interesting thought. I hadn't thought about that. That could be for the bees' perspective and the beekeepers, that could be a beneficial tool for keeping the colony from becoming a gold mine for Varroa. See, one of the things that happens, and I'm glad this question was asked because I forgot to mention it. What happens in my colonies that live in small hives when I do the Darwinian beekeeping with a small hive is they fill up those hives with honey in August, and they've shut down their brood rearing long before autumn has arrived, or not completely necessarily, but it's very minimal. And that means that Varroa is out of luck. Without capped brood in which to reproduce, the Varroa can't reproduce. So that's another. So yeah, judicious use of a queen excluder could help in that regard. Though if there's a honey super above the queen excluder, then the bees are not. They'll still have a brood nest for a long time. So it's a tricky thing.
Okay, I've got a question here, and I wanted to add my own to it too. The question is, just how much swarming do you have? And the one I wanted to add myself is when you're talking about the smaller colony or the smaller hive compared to the larger ones, in the UK, we get told that lack of space is one of the triggers for swarming. And yeah, one of the things we do is put a bigger brood on there to increase the space for them. So when you were doing your experiments or your measurements, did you measure whether there's a greater degree of swarming with the smaller hives than with the larger hives?
Yes, I did. And I need to add that to my talk if I didn't include that. It's, yeah, in this, maybe, let's go back, maybe, did I or not? I can't recall the percentages, but it's almost in the small hive treatment. In the small hive treatment, no, I didn't show that. I don't remember the exact numbers, but in the small hive treatment, almost every colony swarmed. It may have been all of the colonies. I'd have to go back and check that. In the large hive colonies, it was just one or two that swarmed. So that's a big difference. And so, and as I mentioned, the swarming is really valuable for the colony as a means of, it contributes importantly to the control of the Varroa. And I guess I have the luxury of that if my colonies swarm, I'm not creating problems for neighbors. So I can see it, whether you're going to be allowing colonies to swarm or not will depend on your situation.
That does tend to be more of a problem in the UK, Tom, purely because we're a far more crowded place than you are in the States.
Yes.
I've got another question here from Judith. In these examples, I presume you're using movable frames and foundation. What part do you think letting bees build their own comb might play? And do you have an option, or do you have an option on top bar hives?
There are a couple of questions there. First one is, do I have a view on top bar hives? Yeah, those are fine. Those are nice. I don't use those because I like the sturdiness of the comb being surrounded by a frame. But yes, a top bar hive is a more natural situation for the bees. The other question was, let's see, it had to do with, what was the first part of that question? I was thinking about the second part.
In these examples, I assume you're using movable frames and foundation. What part do you think letting bees build their own comb might play?
Well, I do let the bees build their own comb, but yes, I do guide them to build their comb in wooden frames. What I like to do though is I do like to let the bees, when I have them build comb, I will often give them an empty frame with wires to support the comb that they will build. I'll put in an empty frame between two combs where the bees have already built their comb. So they can build whatever kind of comb they want, however they want it, but it's within the frame. So that's kind of that's a compromise that I use. And part of the reason I do that is we're required to have our bees inspected for American foul brood. And so we can't keep bees that are not in movable frame hives. Is that the situation in the UK?
And yes, although, I mean, you do get some people using top bar hives and going very much towards the natural or Darwinian beekeeping anyway.
A top bar hive would be okay because you can still lift out each comb, and that would be let you inspect it. But just putting out a box, that would be you couldn't, and the bees just build their combs in the box. That would probably be great for the bees, but would not allow inspections.
Okay. What are your thoughts about the reasons for colonies coping with Varroa without treatment? Are they physically managing the mites, hygienic behavior, or is it the greater swarming behavior creating brood breaks?
It's a combination of those two things. And the evidence that swarming per se has a huge role in controlling the Varroa by imposing a brood break comes from these experiments where where I could see colonies. I had colonies in treatment groups, and some of within any one treatment group, some colonies swarmed and some did not. For example, in this experiment that I'm showing here with the large hives, there were 2 colonies in there that swarmed, and that really knocked down their Varroa levels. So, yeah, the swarming really, it creates that big long brood break in there, and so that really helps. And it also exports a lot of mites. We estimate that about maybe a third or even more of the mite population leaves with a prime swarm. So yeah,
Question from Suzanne: how many generations of queens before you can consider your bees locally adapted?
I don't know, Susan. It may not be a very large number because every time the queen changes, you're getting really, to a large extent, 50% all new genes in your colony. So after a couple of years, if the colonies are swarming, you're probably getting very locally adapted bees. If there's a population of locally adapted bees in your area, if there are beekeepers buying queens from Italy, or in my case from Georgia, then it's going to be a slower process.
Okay, then I've got a question here from David. Have you marked and noted the age of the queens in your colonies? And if so, what is the average age of viable queens?
In some of the experiments where the colonies are not encouraged to swarm, like these ones where the colonies were in these honey production colonies shown here, the queens in those colonies I have seen queens live for 3 years. Some could live longer than that, but usually the colony is swarmed at least once by that time. So I don't really have good data on just what the maximum longevity is. But I certainly don't, in my experience, I have seen queens that start to fail in their egg laying, and I've seen some queens become drone layers. But usually, but I don't really interfere. I don't try to requeen my colonies. If I do, I want the bees to choose which cells they want to use for their new queens.
Okay. I think that's about it in terms of the questions that you can answer for us, Tom. There's a lot of them. Some of the questions here are related to the local UK conditions. For example, about the density of colonies and how do we control that in our environment. And I don't think that's an answer. Those are questions that you can answer for us. I don't think. I think those are very much UK-based questions. Okay. So unless I can see any more questions which have popped up. I've got a lot here saying thanks for a great talk, Tom. Having read Lives of Bees, I will be revisiting it with some additional notes and thoughts made tonight during your lecture. And many thanks for a great talk, Tom. Many thanks for an excellent talk, Professor Seeley. So a lot of people really greatly appreciated your talk, Tom. I just want to say thank you for a fascinating, thought-provoking presentation, which I thoroughly enjoyed. And as we're seeing from the feedback, so did a lot of other people. So I'm most grateful. Perhaps everyone could show their thanks in the usual way, which is difficult to reflect on a Zoom, but thank you very much indeed anyway, Tom.
Thanks very much, Tom.
Oh, you're welcome.
Lots of thanks very much coming up all over the place here, Tom.
Great. I'm very grateful, and I really especially appreciate, Basil, what you said, thought-provoking, because that's my main goal, is to provoke thought, because everybody's beekeeping situation is different. So if this provokes thought about, that might work, or no, that isn't going to work at all, that's fine. That's good. I consider that a success. So I guess I'll sign off at this point, and let me just express my appreciation. You guys are a real class act over there. Very, very great hosts.
We know.
We've learned a lot, Tom, and your input's gratefully received.
You're welcome.
Thanks.
Bye-bye
Good
now.
night.
Or what do you say, toodaloo?
Some people would, Tom, not many.
Thank you.
Thank you.
Very much, Tom.
Thank you very much.
Bye-bye.
Thank you.
Cheers.
Nice seeing you folks.
Thanks, Kathy.
Thanks, everyone.